The government has released the guidelines for genome editing more than 2 years after the draft guidelines were published in January 2020. Genome editing is a precise way of inducing mutations in plants compared to chemical or radiation induced mutation. With this technique, we can knock off or silence certain genes or introduce novel traits through foreign genes. To give an example, in 2019, a team of scientists from Texas A&M University silenced the gene that produces gossypol in cotton. Gossypol makes cottonseed inedible for humans and animals, but wards off insects. The gene was silenced only in the seed and not in the rest of the plant. As a result, the seed could be eaten like chana, Newswire Reuters reported. The US Food and Drug Administration approved it in October 2019. Products of equivalent risk profile must be treated equally. The regulation and risk assessment as per this principle for genome-edited changes in plants should be the same as for naturally occurring ones. Only when novel traits are introduced through foreign genes, as for example the bollworm killing Bt gene in cotton plants derived from an agrobacterium, should the regulation and risk assessment be similar to that of plants that are genetically engineered or genetically modified. Genome editing can be used for crop improvement, enhancement of crop nutrition, and crop protection. To discuss the guidelines, we have with us Dr. Deepak Pental, geneticist and former Vice-Chancellor of Delhi University. Dr. Pental led a team of Delhi University scientists that produced the mustard hybrid DMH11 using genetic modification or GM technology. The hybrid has a higher yield than conventional mustards. The technique itself helps produce more efficient hybrids in a species that is not easily amenable to hybridization. The GEAC, which is the apex regulator for GM crops, approved it for commercial cultivation in May 2017, but even 5 years later, the government has not allowed it to be cultivated by farmers. Dr. Pantel, welcome to this program and thanks for making the time. Dr. Pental, this is a complex subject. To help us understand what we are discussing, can you tell us the difference between genome editing and genetic modification or genetic engineering
Trouble with
technologies?
in genetic engineering, you introduce foreign DNA, a gene from like Bt cotton. You take a gene uh, from a bacterium and introduce it into the plant, and it stays there. In gene editing, you transiently or for some time introduce a gene like the Cas protein, which is going to help you in editing a particular gene sequence, which is indigenous to the plant. And after the job has been done, that foreign DNA is removed and the plant is back to its composition of what it is originally. So here we use foreign DNA only for a limited period. And once it has done its job, it is important to remove it quickly so that it doesn't create more mutations unnecessarily. It has done its job, you remove it, and the plant is gene edited.
So the foreign DNA is used to introduce the enzymes or the genetic scissors that can do the editing. And once that job is done, they are then separated.
Yes, and that's very easy in plants because you can self the plant or you can cross the plant to wild type, and in the next generation you will have your the construct which you use for mutagenizing the gene, for changing the gene, gets removed. It's very easy
So,
process.
so how are these technologies, particularly genome editing, how is it an advance over conventional breeding?
In conventional breeding, we have used mostly, we have done recombination. We take germplasm which has disease resistance, we cross it to our own best variety and try to incorporate disease resistance into our variety. If the disease resistance is not available within the crop, then you can think about genetic engineering, take it from related wild plant and put it into the plant. Now that would be genetic engineering because that new gene will stay in the plant. But in genome editing, once the job of the enzyme is to make a precise cut, once that has been done, the foreign DNA is removed, and therefore it is very much like natural mutations which occur in organisms all the time.
Okay, so the guidelines categorize plants into three. In the first category are plants whose genomes have been minimally modified with a few base pair edits or deletions. Now, bases, for the benefit of our audience, are four types of molecules that are present in DNA that form DNA. And genes contain DNA, and a genome is a complete set of genes present in an organism. Now, you have three categories, as I said. In the first category are those plants in which the changes have been minimally made. In the second category are those plants whose cells harbor a few or several base pair edits. And in the third category are plants with large DNA changes, including insertion of foreign DNA. Now, the whole purpose of the guidelines is to lessen the regulatory burden or the risk assessment requirements. So, do the guidelines do that for SDN1 or SDN2, that is category 1 and category 2, genetically edited plants, or
You see,
you know,
I must,
is the regulatory
sorry.
burden quite onerous?
You see, it was a very appropriate and good decision by the government Ministry of Environment to issue a notice that the gene edited plants will not be after the foreign DNA, after the mutating reagents are out, it will be treated just as normal mutagenesis. Like when we irradiate plants, we just grow them in the field. There has never been any opposition, there has never been any hue and cry about that, although it might have added lot of bad mutations and recessive mutations to the gene pool. But there has been no hue and cry about it. Here we have targeted a particular gene. It's much more accurate. It's much more less destructive to the plant. And you can just take the plant outside after you have removed the construct which provided you that mutation, and then you can test it in the field like you do with ordinary mutagenesis. And that is the spirit behind the notice that the government issued, that the gene edited plants, after their confinement period is over, after the external DNA has been taken out, will be treated like normal mutagenesis process and will be part of the normal breeding procedures. So the government took an excellent decision, but I'm afraid those who have written these guidelines have created a lot of confusion by not being forthright and clear about what data is required to take it out from confinement to the open field for wider testing.
So, what exactly are your objections? Because the guidelines say that the biosafety committee of an organization that is doing the genome editing will regulate the work during the containment phase, and also whether it can be moved to an open field if there is no exogenous or foreign DNA. Isn't that quite sensible then?
If it was in reality there, actually there is so much ambiguity in what data is required that this defeats the very purpose of the government changing the rules on biosafety of gene edited plants. You cannot leave, if you want IPSC to take decision on when it can go in the open and will not be under any GE rules and regulations, then the guidelines should be very clear. They should be absolutely clear on what data is required. And that is where the whole document is lacking very badly, because it leaves many things up in the air.
Like, for example, the guidelines are talking about stability of mutations. Now, what is ambiguous about that particular requirement?
Once a mutation has occurred, what makes you believe that it will be unstable? If you have found that the plant is homozygous, that both the mother and father inherited alleles are mutated and same mutation is there, then you can self it for so many years until unless a new mutation occurs through natural processes, it is stable. So what do you mean it should be shown to be stable for many generations? This seems to be not much scientific thought that has gone into while framing these guidelines. It makes
So,
things more
sir,
complicated than they
if
are.
I may interrupt you, what is the difficulty in showing that that particular mutation is stable for many generations?
Supposing I am asked, nobody suggests how many generations. That's the number one weakness. And supposing I am asked arbitrarily by the committee that show it for 6 generations, we lose 6 years. So the very purpose of fast tracking and competing with the world in developing new varieties and new materials is defeated.
You said there was ambiguity regarding the data that is required to be shown. What is the ambiguity there? What kind of data is required? Do you know what data is required to
For
be shown?
example, it asks data for non-target mutations. Now that's like looking for a needle in a haystack. The genome is so big, like mustard genome is about 1000 MB megabases. Now you want to find out some 20, 30, 40 or 100 nucleotide sequence in that. It may not be possible. Uh, you have to have certain size and certain length to pick it up even. And what makes you believe that off-target mutations will be always deleterious? And we have already seen that in normal mutagenesis, which we have been practicing for last 60, 70 years, there are off-target mutations. You remove the material which doesn't look good. So when the breeder is going to put their SDN1 plants in the field, if there is anything deleterious or which is not giving the plant giving us the desired results, it will be removed, just like in normal mutagenesis.
So, are you saying that these guidelines are unnecessarily cautious?
They are unnecessarily complicated, and it seems to me that the committees want to be the final decision makers on case to case basis, which is a very bad idea in regulation. Even if you are asking for more data than required or than what is being asked for in the United States or in Australia or in other countries that are allowing easier passage to genome edited plants, you should spell it out. What is it that you want? Leaving it that the developer should prove. Now you can keep on raising the bar and enjoying the scene and destroying the any enthusiasm to work in the area. So it is like committees are gods and developers are at their mercy. The guidelines should be very clear, crisp and clear, which they are not in this case.
Now, if an organization's biosafety committee, the IBSC, rules against a particular applicant, is there any course of appeal?
You see, that is not there. It is not even given properly what is IBSC's role in allowing the movement from confinement to the open situation. When does RCGM step in? This is very clear, as it with the genetically engineered plants, that IBSC records everything and provides time to time the data to RCGM, and when necessary, it goes to the GAC also. That is acceptable. But who is going to rule? And given these complexities in the in the way in which the document is structured, IBSCs will never be able to make any decision. They will
For the
refer it
purpose,
to RC.
for the benefit of our audience, the RCGM is the review committee on genetic modification. Sorry, the review committee on genetic manipulation, which is present in the Department of Biotechnology. Now, sir, you referred to the United States. In March 2018, the United States Department of Agriculture said it does not regulate or plan to regulate plants developed with new breeding techniques, including genome edited ones that could otherwise have been developed through traditional breeding techniques, so long as they are not plant pests or developed using plant pests. Now, do you think that our guidelines are in alignment with those of the United States?
No, no, not at all. The idea behind government's decision was to make it easier, like some of these developed countries which are already started to benefit from the technology. And our scientific experts who have written these guidelines are moving in the other direction. And what is motivating them to make it more complicated, I don't understand. Either gamuts of the technology, or they are interested that they should be the final arbitrators of whether something should go to the field or not.
So, do you think that with these guidelines, our companies, our organizations will not be able to advance in the field of genome editing? You think it's going to be a non-starter like in the case of genetic modification?
Vivian, if you are not clear about what data you are seeking, then arbitrariness comes in. Then it is bad regulation. And that is what it is. So therefore, companies will be very wary of putting their resources into doing something which they are not clear about what data requirements are to be met.
Yes, in the case of genetic modification, our regulations are so difficult, so complicated that many companies that had set up agri-biotechnology laboratories or work or departments in our country, they are pulled back. You think the same thing is going to happen with genome editing as well?
You see, what bothers me the most is that here the government has taken a very good action. They gave a clear notice, and we are messing it up by prescribing data and leaving it loose-ended and not suggesting what exactly needs to be done. So it is some of the members of the scientific community destroying the chances of country doing well in the new and absorbing new technologies and doing something creative with them. So it's a tragedy actually, if you ask me.
But why have they done it? Because they have had the benefit of 2 years. In January 2020, the draft guidelines were published. There was a 1 month window for public comments. I'm sure you also gave your comments at that time. Were you called? Were you consulted when these guidelines were finalized?
No, I gave some unsolicited inputs. I don't know whether those were considered or not, but that is not important. One person's recommendations. What is important is there clarity in the document. And the National Agriculture Academy people got together. They worked very hard. They gave a set of recommendations to the government, to the Department of Biotechnology, Department of Environment, Ministry of Agriculture, all these three departments. And it is very clearly says that when you show that the mutating apparatus is not functional and it has been segregated out and no longer the nucleases or nicases are active, then it should be treated like the way you treat the normally mutagenized plant material. And you pick up the best out of the lot that you have got. This goes totally contrary to that spirit. And it defeats the government's idea of competing with the countries which have already made genome editing much easier. Even countries like Japan, which rejected genetic engineering technologies, have released materials which are gene edited, genome edited. And Britain is, after they have got out of uh, EU, are also reducing the biosafety requirements so that their scientists can produce something valuable for their agriculture through genome editing.
Mhm.
I
Dr.
think this
Pental, it is very
question
sad
was
to know
for me.
that
Yeah.
Dr. Pental, it is very sad to know that the guidelines that have been published, instead of facilitating scientists in genome editing, might actually hinder their work and also act to the detriment of the national interest. For speaking to us very candidly, Dr. Pental, thank you very much.
Okay. Thank you. Thank you.